Great, thanks, Marie. So wanted to just take a moment here and familiarize everybody with who ILSI North America is and what it is that we do. We are a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health. We do this by bringing together scientists and experts from government, academia, and industry to work cooperatively on big emerging areas, including topics like gut microbiome, diet, and health. ILSI North America does not lobby, and we do not develop policy positions. Rather, we are deeply vested in our core values around collaboration, transparency, public benefit, and scientific integrity. To that end, I want to highlight on the right-hand side of our screen that last year, at the end of last year, we were able to earn the Platinum Seal of Transparency from GuideStar. ILSI North America was able to earn this because we were able to not only demonstrate our nonprofit commitment to the betterment of public health, but provide metrics and demonstrate progress on how we were accomplishing our goals and our ambitions of providing science that helps inform confident decisions by all sectors. So with that, before I turn it over to Marie and to our esteemed presenter, I want to thank all of you for taking time out of your schedules. I want to thank ASN for their support on this. I want to highlight that you can reach out to either myself, Gwendolyn Jones, or to Marie at the emails listed. And of course, encourage you to visit our webpage at ilsina.org and follow us at Twitter and on LinkedIn. With that, Marie.
Thanks, Gwendolyn. And with that, I'd like to introduce our presenter today, Dr. Alfonso Benitez Paez. Dr. Benitez Paez is currently a researcher on microbial genomics in the Microbial Ecology, Nutrition, and Health Research Unit of the Institute of Agrochemistry and Food Technology, and that is part of the Spanish National Research Council. And he's been in this position since 2014. In 2010, he received his PhD in molecular biology, and from 2014 to 18, he was a member of the EU project My New Gut, which many of you have probably heard of. Dr. Benitez Paez is also an associate editor of Frontiers in Nutrition, specifically the nutrition and microbes topic area. We greatly appreciate his taking the time to share his insights today, and with that, I will turn the platform over to you, Dr. Benitez Paez.
Hi, everybody. Thank you, Marie, for the introduction. Thank you for the invitation to be in this virtual space free of coronavirus. So to talk all to you about the health fix about diet microbiota health interactions. This is briefly the agenda description for this webinar today. We will review briefly concepts and definitions about gut microbiota and the influence of diet in this particular component of our human body. We also review early microbiota studies, really short, and the methodological basis of the study of human microbiota. We also review the most important aspects and facts retrieved and disclosed by population level studies. We also review the current association between the gut microbiota and different human diseases. And the number 5, we're gonna present the different strategies based on diet for modulation of gut microbiota. Finally, we'll discuss briefly and review the trends and future direction of this research field. As I mentioned, just briefly definition, and just to highlight that microbiota is a common concept to describe all the microorganisms inhabiting a particular ecological niche, for example, human body. This implies the bacteria, archaea, protists, fungi, and viruses, different microorganisms living like commensal parasites or pathogens in a particular ecological niche. Please don't call it flora because it's completely obsolete and deprecated terminology. Not even microflora, it's completely grown. So we have to mention as a microbiota. Concepts. Probably most of you know microbiota concepts. You are related with different methodology to study microbiome from the transcriptomic point of view, proteomic, the pangenome. Also, probably you are familiar, but I want to just to focus in this particular terminology, these biases that you probably find regularly in many scientific articles. The problem with this terminology is completely ambiguous. There is no consensus definition for these biases. So this is ground to refer from my point of view, it's ground to refer these biases in a just ambiguous manner, because just to compare two study groups and to declare that one of them has these biases, just to compare with some different with another control or another interventional group is completely unacceptable. So please avoid to use this ambiguous terminology. About this tree, also briefly, because I want to focus in another more important issues. Just if you want to review briefly the history in the human microbiome research, please visit this important link in the Nature journal that published the last year, summarizing the main milestones in this field of research. So you can see very attractive issues and wholly the full. And finally, I want to highlight the main technology to study human microbiota right now. This is probably you know that most of the study are focused to analyze the bacterial 16S ribosome RNA gene. So this technology is probably the most used because what is cost effective in the terms that they can say who's there in the sample that you are analyzing with a very low price per sample. So probably the main condition to spread the spread of this technology across all the studies in human regarding with the human microbiota. So to enter in the more important issues about the gut microbiota research field and interaction with the gut and other environmental factors, I want to present you some of the most relevant results about studies at the population level regarding the distribution of the gut microbiota across different populations and the association with particular issues and covariates of this important data. First of all, the importance of this kind of level are enclosed to and permit the common microbiota or assess the common microbiota profiles, and despite the great individual specificity variability, because probably you know that the microbiota data is individual specific. The variation across the one population is very high. So this kind of studies permit comparing between population get particular signals inside of the population or inside of the particular group you're interested in studying. Obviously, most of the study, this level comprise the assessment, particularly in gut microbiota, because it's the most extensive type of microbiota analysis right now. also permit to determine the influence of different factors, not only human genetics, but also the environmental factors that are the more strong influence on this kind of data. We also, this kind of analysis or approach also permit correlation of particular dietary patterns with the gut microbiota structures. Essentially, just for an example, the dietary fiber is one of the major dietary components or macronutrients used to modulate right now the gut microbiota, because the links between the capacity of fermentation by the gut microbiota, complex polysaccharides, oligosaccharide present in the human diet. And also compare across population the difference between the countries according to the dietary patterns of different anthropometric or genetic background. One of the major results of this kind of description at population level or including large cohort analysis is the complete description of all microbes inhabiting the human body. No matter the tissue, the cavity that we are analyzing, almost all the microbes inhabiting in our body are already described at different levels, not necessarily at the species level, but we have already reliable information regarding the main genus, families, and phylum inhabiting any particular tissue cavity of the human body. For example, the oral microbiome is completely known. Almost all the species present and associated with particular diseases such as cavities, caries, periodontal diseases are completely known. The gut microbiome is probably the most known ecological niche associated with the human body. We know that the diversity in this particular cavity is huge in comparison with another human tissues. So the greatest scale of the human studies have permitted to describe almost entirely the repertoire of microorganisms inhabiting in a symbiotic manner with us. In such a manner, important human microbiome consortia, also including the European and the North American consortium, that are the two most important around the world, have made important attempts to compile all this information recovered for large cohort and population level studies, just to summarize, or just to understand, and just to have a clear magnitude of the diversity in our body regarding the microbiome composition. At this issue, the last initiative was the International Gene Catalog, published in 2010, I think. Yeah, 2014. We have the reference. And this international catalog compiles almost 10 million different genes described to be present in the human body and associated with the microbiome. So the presence of this kind of different features is huge, tons, several folds more than the information encoded in our own genome. And what about the functions encoded in this metagenome associated with the human? So probably the additional to the conventional central metabolism features that have to be encoded in this kind of genetic information, it's important to highlight that almost 10% of the gene functions encoded in this complex genome, this complex structure, the microbiome, is reserved to carbohydrate metabolism and transport. So this highlights the importance of this particular metabolic function in the microbiomes inhabiting in our body, because this is the main carbon source that they take for the maintenance, reproduction, and growth. Another important fact from the last study, a large cohort study across the world, is that the biodiversity associated with the gut microbiota in this particular case seems to be unlimited. All the years we have reviewers on new papers summarizing the comparing different cohorts across the world regarding particular diseases or even in healthy populations. And the last year in publishing Cell was the largest analysis of the metagenomes across the world. We're talking about the more than almost 10,000 metagenomes analyzed across different populations of the world, and they were able to reconstruct almost 5,000 microbiome genomes at the spatial level. So, and the biodiversity at this level seems to be unlimited, because the more metagenomes you analyze, the more diversity you get. So it seems that one of the facts of the microbiota associated with human is the unlimited biodiversity. And in future studies, probably if you include 20,000, for example, right now the 1 million metagenome project that is ongoing here in Europe and in several countries across the world will define a lot of biodiversity and described previously. One of the key studies in the last year was the description of several covariates that explain the diversity of the human microbiota. In the first years, we were aware, in the first year of the investigation, we were aware that the microbiota associated with human has different factors that strongly influence the structure of the community itself. But in this publication, delivered in 2016, a new covariates were described in order to better understand the factors, essentially the environmental factors associated with the human microbiota. So from this point on, some of the important covariates associated with the human microbiota, and particularly with the gut microbiota, such as BMI and diet and age and the gender, has to be well controlled in the microbiota assessment in order to provide accurate comparison between groups, avoiding the influencing on confounding factors. Another important feature of the human microbiota is the influence of environment factors has over there. So the best exemplification of this is probably these studies in traditional population when compared with the westernized societies, where the dietary patterns difference between both population are completely reflected in the structure of the human gut microbiota. In this particular case, the assessment of traditional populations such as hunter-gatherer of Tanzania exemplifies the enrichment of in gut microbiome that fits with the dietary pattern of these particular people in such a way. We have a predominant presence of, for example, Prevotella species that are enriching in genes deserved to complex carbohydrate metabolism, and it's completely different than microbiota observing westernized population, where the main dietary pattern is based on fat-enriched diets and refined sugars. Additional to this, to reinforce the influence of the diet on the human gut microbiota is the study of seasonal cycling and this particular species. If you take a look in this particular plot, you have the change in the gut microbiota community across the different seasonal cycles that suffer this population and influenced directly by the environmental factors. So you have the strong changes in the gut microbiota community depending if you have the dry season or the wet season, where the sources of food are quite different, because in wet, for example, you have more available of fruits, small fruit, and in the dry season you get more availability of, you have to change the dietary patterns to utilization of animal protein. Another important aspect assessed in the last year and regarding the human microbiota is the transmission, the vertical transmission of the human microbiota between mothers, a child, and offspring. So the studies are using very large cohort and across the population have determined that the mother microbiota is vertically transmitted to the offspring and serves as a seed for development of the gut microbiota, and not only the gut microbiota, but also the oral microbiota and the skin microbiota in the offspring. And you have a particular assessment regarding obesity in metagenomics that is able to distinguish at the species and the strain level, have provided important results at this regard, because mother and children have pretty much the same strains present in the gut and in the skin. So this is a proof in favor of the vertical transmission between mothers and children. And depending on the species transmitted in this couple of individual, obviously, the development of the microbiota in the children will be strongly influenced by different factors associated with the mother health. So it's not strange to have association between the diseases in the offspring, to take a look for diseases in the offspring associated with different mother factors. We also have recovered important relationships between the human microbiota and particular species. And probably the most studied are those diet-related diseases that have unexplained etiology. One of these is obviously obesity, and this is my particular research of investigation. And the very early analysis of the gut microbiota in obesity have retrieved very important results in the way that the obese people seems to have a very low diversity regarding with lean people, so with controls. And this diversity in terms of species and genes present in the gut microbiome particularly has a particular meaning in the conception of the relationship between species in the gut and the interaction with the host. Because as you probably know, the gut microbiota is able to provide effector molecules for the colonocytes, such as short-chain fatty acids. The main of that is the butyrate, but also propionate and lactate. And particular species such as bifidobacterium, Akkermansia play a very important role to maintain the gut barrier homeostasis. In the obese people, all these mechanisms supporting the gut health are completely impaired, and new potential pathogenic bacteria are present in the gut microbiota of these people, altering the equilibrium of all the species present and allowing the development of different harmful conditions such as inflammation by LPS and something like that. But not only obesity has been evaluated in depth regarding the gut microbiota, but also another important diseases associated with this first condition, such as type 2 diabetes, in which, in this case, the main relevant issue was detection of the impact of the drug for type 2 diabetes in the gut microbiota itself. But not only this kind of impact, for example, the metformin, but recent studies have described that non-antibiotic drugs used regularly for treatment of different human diseases have a strong influence in the human gut bacteria, and sometimes inhibit the presence of very critical species for maintaining the gut health. So this kind of studies have paved the way and open a high way of investigation for the impact of different treatments associated with completely different maladies, different than diet-associated maladies in the human microbiome and by extension in our gut health. I also want to describe briefly the gut microbiota studies in the frame of the Dietary studies. And particularly, this kind of cohort is the type 1 diabetes in children. And the studies of the gut microbiota probably have no very clear association with particular species with disease. But at the functional point of view, this consortium have shown very interesting results regarding the composition of the microbiome and the functional level. And they are highlight that the children with developing this disease has low gene reserves to short-chain fatty acid biosynthesis. Different covariates are also associated with the development of disease. For example, the breast milk feeding is a very critical factor in the development of this kind of condition. The colorectal cancer is probably one of the studies better assessed in terms of the influence of the gut microbiota and the evolution and the onset and evolution of this kind of diseases. These results came from different approaches, individual approaches in different cohorts, but robust information or robust evidence came from the metagenomic and meta-analysis carried out in different cohorts across the world. A couple of studies published the last year in the same journal, Nature Medicine. A couple of teams were able to recover unified signals regarding the particular species associated with this colorectal cancer across the different human cohorts. And the results in terms of the pointing out particular species such as Fusobacterium and Collinsella are quite interesting regarding that by different approaches and taking into account different population data, they were to pointed out the same microbes with a strong evidence to influence the development, the onset and development of this disease condition. We have also assessment in IBD and IBS maladies, but not only pointed out a particular species, but also declaring the difference in metabolism composition in the samples obtained from controls and for example, patients suffering ulcerative colitis or Crohn's diseases. Particular metabolism size sphingolipids are quite high in the Crohn's disease compared with controls, and also in the ulcerative colitis. And this particular metabolic signature can be associated with particular species in an indirect mode. And also the association with the irritable bowel syndrome, where several genus and species are are associated and are being disclosed to be depleted, and in particular cases of Crohn's disease, for example, the blue points indicate the species depleted in every single case. Just to review, or just to enter in the particular research field in where I work in is the dietary-based strategies for modulation of the gut microbiota. So probably you know that the non-digestible fiber is the main fuel for gut microbiota, and in such a way, this is the preferred macronutrient used in the different clinical trials to modulate the gut microbiota in order to exert healthy effects on the gut health. Also, it's based on the large epidemiological studies that indicate that sustained intake of dietary fiber protects against obesity, type 2 diabetes, and against cardiovascular diseases. In the year 2016, we published a review in Transient Food Science and Technology, where we review a lot of clinical trials using different dietary fiber components such as whole grain, unily fructooligosaccharides, oligofructosaccharides, GOS, AXOS, ZOS, and resistant starch. And all of them present a common signal or common impact in the gut microbiome, that is a particular bifidogenic effect of the fiber in the gut microbiome. And sometimes this bifidogenic effect is not translated in the amelioration of different metabolic parameters regarding with obesity or metabolic syndrome and analysis in this particular clinical trials. But the different correlation between important species such as Plautia, Eubacterium that are recognized butyrate producers are correlated with the improvement of plasma glucose of even in the inflammatory markers. It shows. So although the beneficial effects of fiber are not directly observed in many times in the different metabolic parameters, the influence of fiber in the gut microbiota through the bifidogenic effect indirectly the bifidogenic boost in the gut can influence another species that are quite associated with the metabolic health in humans, such as Fecalibacterium prosnesi and the other species of Eubacterium, through cross-feeding mechanisms. In such a way, we understand from the dozens of studies that butyrate is probably the main effector molecule resulting from the dietary fiber consumption in humans, because the gut microbiota is able to fermentate this fiber coming from different plant origin, and the direct production of butyrate has different impact on the health at different levels. First one, the presence of butyrate in the intestinal lumen produces a PhD decay in the lumen, so avoiding the expansion and growth of potential pathogenic bacteria and reducing the LPS containing bacteria that can translocate it to the epithelium and producing inflammation. In other levels, the activation of particular receptors in the epithelium cells at the colon are able to produce endocrine molecules regulating satiety and energy expenditure. But not only this kind of function are important at the brain function level, but also to control the insulin sensitivity in pancreas, the lipid synthesis in the liver, and to control the immune response in this particular case, and also to control the beta oxidation and fat storage in the adipose tissue. So as you can see, the consumption of dietary fiber have many many possibilities to at the point to control different mechanisms to maintain the gut health, not only the gut health but at the health at systemic level. I just want to mention briefly results that some clinical trials that we have performed in the latest years. So one of them was the crossover intervention, taking into account the AXOS and PUFA interventions by administration of these compounds during 4 weeks in separated population. We also were able to observe a very strong influence of AXOS in the gut microbiota community because the shifts in this kind of structure, and particularly caused because the influence of AXOS on the bifidobacteria that were the major responder of the AXOS consumption, but not only bifidobacteria, but also Glaucia and another important bacteria producing, able to produce butyrates, such as Staphylococcus and Eubacterium halal, and different species of Glaucia, also Fecalibacterium prosonesi, that are species regularly associated with healthy status. We didn't see any impact of PUFA in the gut microbiota, probably because the short-term interaction with the microbes. But in another randomized trial performed by other authors and exposed the gut microbiota to long-term intake of PUFA, they were able to observe similar changes to that observed in our study with AXOS. In such a case, the bifidobacteria, Succinivibrio, Silibabrio, and many others butyrated producers were able to respond positively to the PUFA intake. But not only the fiber is able to modulate the gut microbiota, but also the caloric restriction itself is able to modulate in an important manner the gut microbiota and to reproduce and to translate into the more visible changes at the adiposity and metabolic level. In a recent clinical trial using calorie restriction in combination with fiber supplementation, we see a very strong influence of calorie restriction, particularly in most of the anthropometric measures, as well as in the lipid profile and glucose homeostasis markers. But and particularly the interaction between calorie restriction and fiber was observed in the control of blood pressure, because both markers of systolic blood pressure and diastolic blood pressure were improving only when the presence of calorie restriction and fiber. And this study was also important because apart from the influence of diet and time exposition of fiber to the gut microbiota, we recovered that sex was a very important factor influencing the gut microbiota and influencing the amelioration of different markers analyzed for this cohort. And globally, we observed that the presence of fiber in the calorie restriction regimen was influencing positively the abundance of Parabacteroides, a member of the Bacteroidetes phylum that is recognized by enriching in a lot of metabolic pathways associated with degradation of complex polysaccharides, as well as obviously the expected bifidobacterium or bifidogenic effect of the fiber, and interestingly the reduction of bilophila species associated with the inflammation in adipose tissue. We also have associated important the change in the phenotype from the lean to obese phenotype in children. And we were able to distinguish particular microbiota communities or particular gut microbiota signatures associated with the maintenance of the lean phenotype across the time, and in comparison with those communities associated with the transition from lean to obese phenotype. And particularly, this kind of microbial structure associated with the transition between lean to obese phenotype were correlated with the development of inflammatory markers in this kind of in this population. So probably the link between these communities and the inflammatory markers are very strong. And we also retrieve association not only between these kind of markers, but also in response to the dietary patterns characteristic of these different groups of children. In those, we discovered that the intake of in particular, the simple carbohydrate or refined sugars were high in the population or in the children with transition between lean and obese phenotype. And the consumption of fiber was inversely correlated, being higher in the lean maintaining phenotype children. Finally, I want to introduce the current trends and future direction in this kind of research field. And one of the main fields to investigate in the next year is the strain level variation and the gut microbiota. It's necessary to refine the tools and the technology to describe the microbial community at the strain level, because this will permit to determine better association between microbe and host with the diet. This is a very nice example published the last year by the Nicola Segata group in Trento University in Italy, where they discovered the variability of Probiotica copri. The species is strongly associated with fiber consumption, and they are able to disclose that even vegans, vegetarians, and omnivores contain different Probiotica copri species. And this is reflected directly in the functional composition of every single strains associated with every single group. Another important key issue is the integrative biology in the next years. The integrative human microbiome program was started months ago, and right now we are developing a lovely study combining different multivariate data. And this in such a way, the studies in the next month or next years or next month even will combine very complex information, not only regarding microbiome assessment, microbiome surveys, but also information regarding proteome, metabolome, lipidomics, clinical data, epigenomics, and all this kind of information has to be compiled in a very comprehensive manner to disclose very most robust interaction between microbiome and diet and host. Examples of this kind of interaction have published very recently in the case of multi-omics assessment in inflammatory bowel diseases. Take a look for all different data integrated in this study, and the results are quite exciting in such a way that not only taxonomy of particular microbial species are associated with disease, but also bile acids and transcription factors could be get insights into development, onset and development of this disease. We have published recently the multi-omic assessment in the AXOS intervention, and all the information regarding metagenome, lipidomics, and metabolomics converge in a one unique signal is the improvement in the glucose homeostasis in these overweight patients. But not only multi-omics, but also the development of next generation probiotics. Probably you must know the different studies performing in acromantia associated with the obesity and improvement of glucose homeostasis in humans and animal models, as well as Eubacterium halai. In our laboratory, we are developing different next generation probiotics that are distinguished because they are not the classical lactobacterial acid regularly used, such as lactobacillus and bifidobacteria, but are different and correspond with the most abundant species present in the human gut microbiome. We have performed assessment to provide the glycolytic versatility of this kind of bacteria with very interesting results regarding bacterial uniformity. We have provided safety assessment using this particular strain, demonstrating that it's safe to be used in future human clinical trials. And interestingly, these results in animal models describes potential metabolic interaction with another important species associated with the human health, for example, Christensenella and Akkermansia species. And just to highlight, fecal microbiota transplantation, classically used for treatment of Clostridium difficile infection, but also right now there are current trends of using this kind of therapy to treat ulcerative colitis and other IBD conditions. Just to close, concluding remarks, just to mention briefly, the microbial diversity inhabiting the human body seems to be limited because the more metagenome, the more analysis you perform, the more diversity you find. And the causation in human microbiota for most diseases is explored, is not constantly supported. So more mechanistic functional analysis required to provide a direct role of human microbiota in different diseases. And diet and lifestyle are the major drivers of gut microbiota shape, but this kind of interaction between bacteria, the diet and the bacteria can mask another kind of interaction, another interesting interaction between different species of the bacteria because of cross-feeding. And regarding the result of the next probiotic generation, are dead or attenuated probiotics only necessary to exert metabolic change? We have to study more in a more mechanistic way if all these kind of issues have immunology behind of this kind of dysfunction. And also regarding the next generation probiotic, health claims of single probiotics provide robust evidence to be approved for human consumption. In this such a way, we had to expand the QPS status for a broad number of human symbionts, not only for lactobacillus and bifidobacteria. And microbiome database results lack of experimental evidence supporting functional prediction. And this kind of analysis or studies are the immediate objectives for the next decade. That's all. Thanks for your attention. I'm happy to answer your questions.